I recognize some of the people coming in the room. Oh my gosh. I'm waiting for the waiting room to empty.
230 participants.
So far, they're still streaming in here. Hi, Diane.
Hi, how are you doing?
Good. Okay. Great. So while people are still entering from the waiting room, I'm Carla Neugebauer. I'm the director of the RNA Center at Yale. And I want to welcome everyone to the very first seminar of the RNA Collaborative. We're a collection, as you may know, of 7 different RNA centers. And this was really, as far as I know, spearheaded by the admins who recognized how stressed out all of the PIs are around the world, and that they couldn't possibly, get their act together and organize something like this. So they've done us a fantastic favor of putting us all together and having us volunteer to give talks that everyone who's deprived of their beloved molecule RNA and their excitement about science can join us here and listen to really great talks every 2 weeks. So I hope everybody knows that the idea here is to have these talks every 2 weeks, and the different locations are putting forth the speaker list. So I wanted to acknowledge the admins of the 7 RNA centers that have collaborated to come together to put together this RNA collaborative seminar series. I want to also say that we're going to have 2 talks today, both from Yale, and what I'll do is use the chat room to, I see there's some chats already in there. Great, practically a conference. That's right. We're getting up north towards 300 already. So if you have questions during the talks, because I know that during talks over Zoom, you lose that audience atmosphere, which is unfortunate. But if you have questions, you can either put them in the chat, and I can ask Sarah or Nadya after the talk so that we can have a lively question and answer. You can also raise your hand, and I can help the speaker by calling on you, and you can ask the question in your own way because you might do a better job than I do. So we'll manage. We're a friendly group here. I think we all know that, and so let's use those 2 different tools. I think everybody knows about raising your hand in the participant section. If you click on participants, there's an option to raise a little blue hand. And then there's the chat option down at the bottom, just as a reminder. Okay, so I'm going to introduce the first speaker, who's Sarah Slavoff. So Sarah is an assistant professor in the departments of chemistry and molecular biophysics and biochemistry at Yale. She started her lab here in 2014, having done a Ph.D. in chemistry at MIT with Alice Ting, and a postdoctoral fellowship with Alan Sagatilian at Harvard University. So she was involved in identifying teeny tiny proteins, which I've also always found extremely fascinating about her work. And then this has led her towards looking at RNA in cellular subcompartments. And so without further ado, I mean, I'm looking forward to her talk. I do want to tell you that she's a Searle Scholar and that she's off to a great start in her lab at Yale. And so I'm going to turn myself onto mute. Everyone should be muted, but during the question and answer period, you'll be able to unmute yourself to ask your question. Okay, take it away.
All right, thank you so much, Carla, for the introduction. And really thank you, Carla, for organizing this entire event for us here at Yale, and also for being the person who dragged me pretty much single-handedly into my involvement in the Yale RNA Center from the time I started here. I credit you with sort of a lot of influence over our work here. So what I'll be talking about today is regulation of the specificity in the process of RNA decapping, which is an early step in RNA degradation in the cytoplasm, and also of phase separation and formation of cytoplasmic RNA protein bodies by a small human polypeptide. First I want to just start off with some background on these little polypeptides that Carla mentioned that my lab specializes in studying. So we work on really nothing less than the dark matter of the human genome. So it turns out that small open reading frames, also sometimes rather cutely abbreviated SMORFs, in the human genome were previously invisible to geneticists, as you can see illustrated from this graph from almost 15 years ago now. Annotated mammalian proteins, when plotted by size, exhibited this dramatic but actually artifactual drop-off when you got down to 100 amino acids. And that's because genome annotation consortia implemented this arbitrary cutoff, because as you get to shorter and shorter lengths of open reading frames, right, sort of statistically, the odds that they will appear in the genome become quite large, but it's then difficult to differentiate which of these small ORFs are genuinely coding and which are not. So we ignored them, and then we forgot that we were ignoring them until recently, when really sort of the first breakthrough in this field was the invention of ribosome profiling by Ingoglia and Weissman, which revealed translation of thousands of small open reading frames in the yeast and then mammalian genomes. And then labs like ours, while I was a postdoc with Alan Sagatilian at Harvard, developed proteomic methods to combine proteomics with RNA deep sequencing to reveal these translated small open reading frames. So to give you sort of a brief overview of how that proteomics method works, any sort of sample, it can be microbial cells, it can be human cells, it can be tissues, is lysed and then subjected to some fractionation method to enrich small size proteins and peptides from the sample in order to increase detection sensitivity, followed by a standard trypsin digestion and mass spec workup. The difficult part comes in the bioinformatic analysis, because normally proteomics works by matching obtained spectra from your proteomic sample to spectra derived from annotated proteins. But of course, that doesn't tell you about anything non-annotated. So we translate all of the cellular RNA in 3 reading frames and eliminate any biases to start codons as well, which was unexpectedly informative. And then we're able to match all of our spectra and eliminate matches to known proteins in order to derive detections of new non-annotated sequences. So there have been a number of surprises in this space, and I now feel brave enough to use this term human operons in describing some of these small genes because John Weissman coined it in his recent science paper. So as a matter of fact, we see that small open reading frames don't just break the rule about the size of human genes, they also break other rules. So for example, they're enriched in non-AUG start codons relative to the proteome as a whole. Additionally, they are encoded in non-coding RNAs, which were sort of misannotated as non-coding simply because they are translated into small proteins, but also in mRNAs along with known functional proteins, upstream, downstream, and in overlapping frame shifted open reading frames. And in John Weissman's recent large-scale genomic CRISPR screen, he demonstrated that in fact many of these upstream open reading frames aren't just translational regulators as was once assumed, but are important functionally for cell survival. So the sort of term human operon suggests that 2 functional proteins can be co-encoded in the same human mRNA, which was a surprise. Indeed, many small ORFs have now also been characterized functionally at the molecular level. So here I provide a few examples from my lab in recent years, really just an overview to convince you that many different important cellular processes can be regulated by these small proteins. So we have now a preprint up on the bioarchive describing a novel human small open reading frame co-encoded with a ribosomal protein that regulates the activity of a lipid transferase that localizes at ER plasma membrane junctions. We have, and others, many others, have identified small open reading frames that are regulated in their expression and some that are functional in cellular stress responses. And the small peptide, the small open reading frame encoded polypeptide that I'll tell you about today, which our lab named Nobody in our 2017 report, which I hope I can convince you regulates both cytoplasmic 5' to 3' RNA decay at the RNA decapping step, and also the dynamics of these associated RNA protein granules called P-bodies. Just to give you a little bit more background, this complex of proteins called the decapping complex consists of sort of many accessory factors as well as the decapping enzyme, DCP2, which catalyzes the first step in 5' to 3' RNA decay, which is removal of the 7-methylguanosine cap. These RNA protein granules called P-bodies were first described by Roy Parker's lab in 2003. And they're really fascinating because a number of important sort of mysteries remain about them. So they are known to be liquid droplet-like cytoplasmic ribonucleoprotein granules. So, you know, potentially are formed via this now very hot process of liquid-liquid phase separation in biology. They're known to be enriched in deadenylated transcripts that are not yet decapped, as well as the protein components of the RNA decapping complex and associated proteins involved in translational repression and RNA degradation. Now, it's also known that these P-body associated proteins and RNAs are in dynamic exchange with the cytosol via FRAP. But even more interestingly, two very recent single particle imaging papers from the Parker and Chow labs demonstrated that these P-bodies are more like storage granules because 5' end decay of RNA is not occurring inside the P-body. It occurs on these either subdiffraction limited tiny P-bodies or individual decapping complexes in the cytosol. So now that opens several more questions, right? So what is the function of the P-body? How are these transcripts and protein complexes liberated from P-bodies? What are the conditions that regulate dissociation of P-bodies? And of course, from our perspective, what is the role of this small protein, Nobody? So with that context in place, let's start from the beginning, the discovery of the Nobody polypeptide, which was just observed in a proteomic screen of a number of different human cell lines in which tryptic fragments mapping to a small open reading frame in what at the time was annotated as a long non-coding RNA were observed. And the full coding sequence of the Nobody polypeptide was inferred to generate a 68 amino acid, what we'll refer to as microprotein, is a term that has been used in the literature. So the sequence is shown here below, and I hope that you will be convinced that there's significant sequence identity for this gene within mammals. Now, while this sequence is sort of bristling with apparent interesting functionality, enrichment in prolines, which confers intrinsic disorder, which we have shown via NMR, as well as these stretches of very high positive charge density. But it wasn't apparent in the beginning exactly what this peptide might be doing. So it took functional proteomics of co-IP and mass spectrometry, followed by molecular validation, to demonstrate that when you pull down the Nobody polypeptide from human cells, it co-purifies with the components of the RNA decapping complex. And we have done a lot of in vitro biochemistry, which I won't present today for brevity, to really demonstrate that the direct binding is to this accessory protein or scaffolding protein, EDC4. Okay, but inside cells, we now have great antibodies against the endogenous no-body polypeptide, as well as its phosphorylated variants, which I'll talk about in a bit, that demonstrates that indeed this endogenous protein at least partially co-localizes with endogenous P-bodies, here marked by the P-body resident protein DCP1A, which is a decapping accessory factor. We've also done quantitative Western blotting to demonstrate that no-body is present at the same order of magnitude of concentration inside the cell as its binding partner EDC4. So the stoichiometry that we're able to derive from experiments like this is approximately 3-fold more EDC4 than no-body inside the cell. It's also interesting to note that CRISPR knockout of the no-body gene changes the number of P-bodies in the cell. So we see approximately a 3-fold increase in the number of P-bodies in no-body knockout cells, which can be partially rescued by reintroducing the no-body coding sequence. Okay, so endogenous no-body indeed appears to be associated with the decapping complex. Now, we were able to undertake a really wonderful collaboration with our lab neighbors, Matt Simon and his grad student, Jeremy Schofield, to use their time-lapse seq method of RNA metabolic labeling and dynamic profiling to ask what happens to the dynamics of RNA in no-body deletion cells. We would expect if no-body indeed is regulating RNA decapping, that we should see differences in RNA stability when we knock out the polypeptide. And indeed, what these plots are showing you is that we see really dramatic dysregulation, actually both up and down, in RNA abundance. And I should note, these measurements are all on mature RNA, so this is not direct profiling of nascent transcripts. So we see dramatic changes in the abundance of RNA in both directions in the no-body knockout cells. And if, for example, we separate out these transcripts that increase in abundance, we're able to model that a number of them change as a result of RNA decay, and also a number of them change as a result in changes to RNA synthesis. We interpret these changes in RNA transcription and processing as possibly representing genetic compensation for the loss of the no-body gene in cells. And we interpret these changes in RNA decay rates as being related to changes in RNA decapping as a direct result of the loss of the no-body polypeptide. The reason that we feel we can make that inference is, first of all, we have made measurements of the rate of decapping of transcripts that we have determined in other work to be direct substrates of the DCP2 decapping enzyme. And we see that in cells just lacking a downstream exonuclease, which allows decapped transcripts to accumulate so that we can measure them, a splinted ligation assay that specifically reads out the amount of decapped RNA in cell by a qPCR shows accumulation of the decapped transcript in those cells. But when we knock out either the decapping enzyme or just the no-body protein in addition, we lose all of that accumulated decapped product, meaning that at least for this gene, both the decapping enzyme and the no-body polypeptide are required for its efficient decapping in the cell. Okay, so nobody is affecting the decapping step. However, okay, in general, we do see a positive correlation of the change in RNA stability in the no-body knockout cells relative to wild type with a DCP2 knockout cell line relative to wild type. And that was work separately undertaken by a very talented graduate student, Vicky Luo, in my lab. But that correlation is not perfect. We see some DCP2 substrates, for example, stabilized in the no-body knockout and vice versa. And also no-body targets, DCP2 targets, I apologize, that are destabilized in the no-body knockout, okay? So it really sort of just dysregulates the rate of RNA decay of DCP2 targets. So our best interpretation of these data is that what nobody is doing is it's specifying the targets of DCP2. So it's required for correct selection or correct specificity of decapping targets. So what we can take from this is just that overall, deletion of this tiny polypeptide is dysregulating the rates of decay of thousands of transcripts in human cells, and also possibly inducing compensatory changes in gene expression. Now, we were also curious about how and why this small polypeptide appears to be regulating the numbers and dynamics of p-bodies in the cell. And of course, this may be related to RNA decay rates, or it may be a separate consideration altogether. Okay. So let's just treat p-bodies as a separate question for now, and we'll come back to the functional relevance at the end. Okay, so we never really observed any sort of meaningful changes in no-body transcription or protein abundance in the cell. But what we do observe is that under specific conditions already known to regulate p-bodies, no-body can be post-translationally modified. It can be phosphorylated. So in particular, we first looked at mitosis. And there are several papers, for example, from the Slavoff lab, that show that p-bodies disappear during mitosis. And what we observe is that during mitosis, nobody is phosphorylated at a specific position, T40, near stoichiometrically. That is a specific modification. So if we mutate that threonine residue, we lose the phosphorylation. And that phosphorylation is installed by cyclin-dependent kinases, as you would expect at mitosis. What's even more remarkable, though, is that that phosphorylation is required for p-body disappearance during mitosis. So in cells expressing wild-type nobody on the nobody knockout background, just so that we have comparable genetic backgrounds, you see the expected decrease in p-bodies when we treat with an inhibitor that freezes cells at the G2M checkpoint, nicotazole. So these cells are entering mitosis. All of the p-bodies have disappeared, essentially. But if we just express a non-phosphorylatable version of the no-body polypeptide, the p-bodies essentially stay on. There's almost no change. So while this modification is not sufficient to dissociate p-bodies, in the context of mitotic signaling, it is necessary for p-body dissociation. Okay. Now, similarly, we looked at another signaling condition, EGFR signaling, and we observed a different phosphorylation site is installed on nobody. But again, it's highly specific. It is near stoichiometric. And in this case, we identified the immediate upstream kinase as being not EGFR itself, but protein kinase C, PKC, both in vitro with a PKC inhibitor and in vitro. We also observed that this no-body phosphorylation site is endogenously upregulated in cancer cells with mutant EGFR that is self-activating. And a nice control suggested by our own Mark Lemon, for which we thank him, is this cell line has a resistance mutation in EGFR to erlotinib. And in that cell line, we see that inhibition by erlotinib is abrogated, really suggesting that this is specific to this signaling pathway. Although I would expect that any conditions that activate PKC should install this phosphorylation site on nobody, not EGFR specifically. Now, under these conditions, it was not previously reported that p-bodies would dissociate, but we observe very cleanly that treating cells, these are HEC293 T cells, expressing wild-type nobody, induces, again, almost complete dissociation of p-bodies. And also under these conditions, remarkably, phosphorylation of nobody at that single site is also required for p-body dissociation. So when we're expressing the non-phosphorylatable mutant, we can't dissociate the p-bodies. And in this case, we did the control with the phosphomimic mutant of nobody, and we observe, again, that p-bodies are able to dissociate. So I really just briefly want to discuss some in vitro investigations, because this really, it really raised the question in our minds, then does nobody have sort of the direct ability itself to phase separate with RNA? It is itself intrinsically disordered and positively charged. And multiple reports from Christine Keating at Penn State, and more recently in several papers on G3BP1 protein, have suggested that phase separation can be modulated by phosphorylation. So what we observe is that the no-body polypeptide, under specific buffer conditions, when mixed with RNA, can indeed induce the formation of liquid droplets. and that this is dependent on the concentration of the polypeptide and inversely dependent on the concentration of salt, exactly as you would expect for this non-specific phase separation process called complex coacervation, which is formation of liquid droplets via the interaction of oppositely charged polyanions, like RNA and a positively charged, intrinsically disordered polypeptide. Okay, and I think what's particularly satisfying here is that we were also able to show that by incubating protein kinase C in our mixtures, we were able to observe dissociation of these complex coacervates of the no-body polypeptide with RNA in vitro. And that if we perform this experiment instead with a non-phosphorylatable mutant of nobody, that addition of PKC and the additional buffer components does not dissociate the liquid droplets as observed by the decrease in turbidity of the mixture. Now, we find these in vitro results really compelling and really interesting. Their relevance in the context of the cell and the P-body remains to be established. It is possible that the mechanism by which phosphorylation of nobody induces dissociation of P-bodies inside the cell is simply an electrostatic repulsion induced by the introduction of the additional phosphates. It is also possible that this is a much more complex mechanism that would involve additional changes in the composition of the P-body at the protein or RNA level. I mentioned that there will be sort of more interesting discussions related to the functional outcome here, because there's little change in the rate of RNA decay that we observe under these conditions. So the reason that cells want to use nobody to do this process remains an active area of investigation. So with that said, I want to thank the students who did this work, Dr. Zengquan and Vicky Luo, and our collaborators, and I'm happy to take questions.
Okay, so thank you very much. I think we can all use our reactions to clap or give a thumbs up if you feel so moved. Thanks, Sarah, for her talk. So now we'll have some question period. I guess we can have about 5 minutes of questions if they show up here. I'm just looking for a show of hands. Hopefully everybody knows how in the participants section to raise hands. And I don't see questions in the chat channel. So that's the other way you can ask a question, is to send a question in the chat and I can read that out. Here, Nils Walter has a question. Go ahead, Nils.
All right, I'll turn on the video too, out of courtesy. So great talk, Sarah, thanks. I was just wondering, so normally when one thinks about phase separation, as many have described, and us included, you think of a large surface area and multi-valency of the interaction with a small peptide that, of course, runs counter to that intuition. And so how do you think, I mean, what exactly does nobody do to affect changes?
It's a fantastic question. So, in vitro, peptides of similar length with similar high local positive charge density have been reported to undergo complex coacervation. So again, I refer in particular to Christine Keating's really fantastic Nature Chemistry paper from 2016. That really was the motivation for us to think this could happen in vitro. But I don't know, okay, whether nobody even directly contacts RNA inside cells. And I think that's really sort of the key question to answer you more deeply. Because the only way that this in vitro observation would have any relevance in the context of the cell would be if that's the case. It is clear from our results that inside the cell, phosphorylation of additional proteins in the P-body is essential for nobody to have this effect on the P-body. So if you just express the phosphomimic version of nobody inside the cell under basal conditions, nothing happens to the P-bodies. So my current best hypothesis is that, by phosphorylating many P-body resident proteins, we create this sort of, electrostatic repulsion on larger scales, over sort of larger surface areas. And for some reason, this one phosphorylation site on nobody is an essential component of that network.
Great, thanks.
So can I follow up on that? Because that was similar to my question, but then based on your answer, so clearly there's multiple phosphorylation events that have to do with assembly or disassembly of the P bodies. When you use those phosphomimetic or abolishing mutants like alanine, do you see a change in the apparent size or number then of P bodies?
That's a really, really good question, Carla. So we haven't directly quantified P-body size. We're mostly just doing object counting. Is there a P-body there or not? But there definitely are conditions that we have looked at that seem to change the size of P-bodies. It's a little bit harder than some of the really cool nuclear bodies that people like yourself are working on because P-bodies on average are about 200 nanometers, so they're sort of diffraction limited. So I think we might need to do a little bit better microscopy to answer that question as rigorously as it should be answered.
Cool. People I'm seeing in the chat room, people are saying they don't see the Q&A. If you can raise your hand in the participants section, if you click on participants. There we go, Omar.
Nice talk, Sarah. I have a question. I wonder if you have looked at the composition, the RNA composition of these P bodies, and I wonder what would happen if you express another RNA that is misfolded, not structured, like nobody in the cell.
So to answer the first part of your question, there was a really fantastic 2017 molecular cell paper from Dominique Weil's lab that did really beautifully catalog the RNA composition of P bodies via sort of a partial purification using FACS. And I refer you to that paper. In separate work, we've actually found that the P-body transcriptome and the RNAs regulated in stability by the DCP2 decapping enzyme are almost completely overlapping. So I think that's a really good list that they published. In terms of, modulating the RNA composition of the P-body, we haven't tried that yet. So it's an interesting suggestion.
So I see a question, I don't know who it's from, DK59. Do the cells seem to care that they can't decay their P bodies during mitosis? I thought that was a good question.
That's a really, really great question. So that's sort of active work in our lab right now. I mean, there's no obvious, our T40A nobody mutants those cells aren't dead. Okay, so they're not totally dying. But we are interested in asking what the functional consequences of these things are. So, we don't observe any real clear changes in RNA degradation when we perturb P-body formation or dissociation. We're currently asking, inspired by Roy Parker himself, actually, when he was recently at Yale, who asked me if translation is changing. We're also interested in just determining if this is sort of a redistributional effect where the cell needs to send the P-body components equally to daughter cells during division. So those are all good hypotheses that we're following up on right now.
Really great. So I think in the interest of time then, we need to switch to the second talk. I'm seeing participants coming in and out of the room. I hope we're going along without too much of a hitch here. So Sarah, if you can unshare your presentation and then Nadia can come on. Thanks again, Sarah. That was terrific. So I want to introduce Nadya Dimitrova, who's hopefully sharing her screen right now. And Nadya is an assistant professor at Yale in molecular, cellular, and developmental biology. She's started her lab in 2015. And she had done a postdoctoral fellowship at MIT in Tyler Jacks's lab, where she discovered the phenomenon that she's about to tell you about. And she had done previously her PhD at the Rockefeller University with Titia Dillon, and she is a Pew Stewart Scholar. So without further ado, I would like to welcome Nadya.
Thank you, Carla, for this great introduction. And Carla, of course, one of the rare advantages of being isolated at home is the opportunity to keep up with TV programs such as the inspiring news documentary by Ken Burns, The Gene, which was aired on PBS a couple of weeks ago. And so in this beautiful snapshot from the documentary, Burns illustrates the striking observation that protein encoding sequences comprise only a minor fraction, about 1 to 2% of the human genome, and are thus unlikely to account all by themselves for organismal complexity. Instead, they are surrounded by a wealth of regulatory sequences, which are there to modulate gene expression, to determine if a gene is turned on or off, when it becomes expressed, in what cells, and to what extent. And so in the textbook view of the gene, we often think of these regulatory functions being fulfilled by specific DNA elements, such as promoters or enhancers. But it may be worthwhile to consider how non-coding RNAs, the active transcription of a non-coding RNA from a given locus, or the accumulation of non-coding RNA species might be contributing to this regulatory landscape. And when we think about these functions, we are stepping into the realm of long non-coding RNAs. So long non-coding RNAs are a class of RNAs with a very simple definition. They're non-coding transcripts longer than 200 nucleotides. But of course, as you just heard from Sarah, non-coding could be something that could be challenging to determine in some cases. Nonetheless, long non-coding RNAs as a class are found to be pervasively transcribed in mammalian genomes, expressed both from intergenic regions as well as from portions of the genome overlapping protein coding genes. There's likely hundreds of thousands of them in the human genome alone, and they're generally speaking poorly conserved and not very well characterized. And while it may be easy to imagine that these transcripts are simply transcriptional noise, some data indicates that at least a fraction of those, a subset of those, are of functional significance in health and disease states. And so in my lab, we primarily investigate the roles of long non-coding RNAs in the regulation of gene expression in cancer-relevant pathways. And so our interests lie at the intersection of tumor biology and the biology of long non-coding RNAs. And as an entry point, we began by examining long non-coding RNAs in the context of the p53 pathway, which is a central tumor suppressor mechanism, mutated or inactivated in over 90% of human cancer. So p53 itself is a transcription factor, which in response to stress, such as DNA damage or oncogenic signaling, becomes stabilized, and it binds to motifs called p53 response elements in the promoter of target genes to activate their expression and ultimately enact its downstream functions and tumor suppression. And so our goal was to identify and characterize the functions and mechanisms of p53 regulated long non-coding RNAs, both in order to understand their contribution in the context of the p53 pathway, but also as a means to perhaps determine to what extent, what is the scope of contributions of long non-coding RNAs to cancer more broadly. And so to this end, Efrat, a graduate student in my lab, analyzed the p53 transcriptome, and she found that p53 binds to several thousand sites in the genome to activate a couple of hundred genes, and among them about 20% were long non-coding RNAs. And so over the last couple of years, my lab has been systematically going through this list. And in my talk today, I would like to focus on two long non-coding RNAs which emerged from these studies. These were the first two that we characterized. The first one, PBT1B, an isoform of the long non-coding RNA PBT1. This is a study we recently reported on. And the second one is linked P21, which I characterized a number of years ago as a postdoc. And so both LINK-P21 and PBT1B share multiple common features. They're both spliced, capped, and polyadenylated transcripts with mature length of about 3 kilobases. They each have a p53 response element designated here in their promoters, and so they become induced by p53 during stress. Their subcellular localization is also notable. They're both chromatin bound. And in fact, if you look by single molecule RNA fish using intronic and exonic combination of probes, one realizes that that both of these long non-coding RNAs actually never leaves the site of transcription. And what struck us the most related to their subcellular localization was also that we found that both of these long non-coding RNAs were primarily involved in the regulation of their neighboring genes. Specifically, we found our studies reveal that PBT1B acts to repress the expression of its neighboring gene MYC, which is a very important oncogene and sort of a promoter of cellular growth. And we discovered this as sort of a previously unappreciated strategy which p53 uses to suppress cellular proliferation during stress. On the other hand, LINK-P21 has the reverse role. It actually serves to promote the expression of its neighbor gene P21 past the functional threshold, in this way allowing cells to initiate the G1S checkpoint in response to DNA damage. And so from these studies, we concluded that at least a subset of long non-coding RNAs are involved in what was referred to as cis regulation, basically their ability to modulate the expression of their neighboring genes. Now, just to take a step back, historically speaking, this idea that long non-coding RNAs can be involved in cis regulation is not surprising. It's actually been an integral part of the field with examples such as the role of exist RNA in X chromosome inactivation. Here is exist coating, the X chromosome which it silences, and in some other instances, the role of long non-coding RNAs such as AIR in the repression of imprinted loci. And so to further study now what we were suggesting, which was a potentially expanded repertoire of functional cis-regulatory long non-coding RNAs which could be found throughout the genome and which had the capacity to positively or negatively modulate and therefore tailor the expression of their neighboring genes. We have pursued two main questions, which I'm going to talk about today, with the first one being, what is the physiological relevance of cis-regulatory long non-coding RNAs? In other words, if a long non-coding RNA was to be perturbed, would that lead to physiological outcomes at the organismal level? In our case, perhaps some changes in tumor development. And if cis-regulatory long non-coding RNAs are functional, how do we define what are the functional elements within them? What are the elements that define their ability to perform positive or negative regulation? And today I'm going to, the first question we addressed in the case of PBT1B, whereas the second one we're going to focus on LINK-P21. Okay, so to focus on the question of functional significance, I'm going to start with PBT1. And so PBT1 is a long non-coding RNA, one of those old long non-coding RNAs discovered all the way back in the 80s. And it was discovered precisely because of its close proximity to the MYC oncogene. So a number of studies discovered that PBT1 locus, as well as transcript, so genomically and transcriptomically altered in a very large fraction of human cancers, and it's usually correlated with increased MYC activity. And so these findings from the start led researchers to propose that PBT1 has an oncogenic role in cancer. Cristian Olivero, a graduate student in my lab, was however particularly intrigued by the potential for a tumor suppressor function, given the association of p53 with this locus. And so she used CRISPR to specifically mutate the p53 response element, generating this delta RE allele that disrupts p53 binding in this locus in order to ask what would be the consequences of loss of PVT3-dependent transcriptional activation and induction of PVT1B. And so she showed, first by chip, that mutagenesis of this site indeed abrogates P53 binding, which is observed in control cells in the presence of stress. And she also showed that while PVT1A, this is an isoform of PVT1, which is initiated from this exon 1A, an upstream exon, we believe this isoform to be constitutively expressed, that is, it's not stress inducible, and it also doesn't significantly change in the presence of our mutation. On the other hand, PVT1B, which is initiated from an alternative transcription start site, is exon 1B, immediately downstream of the P53 response element. This isoform again becomes induced in the presence of stress by P53, and it's inhibited following the mutagenesis. So next, she also showed both at the RNA and at the protein level that this delta RE mutation did something very interesting, which is that it rescued the downregulation of MYC that was observed in control cells in the presence of stress. So again, just to repeat that, whereas MYC levels drop about 30 to 50% during the P53-dependent stress response, they're rescued if P51B is inhibited. And so these initial findings from the start led us to conclude that PVT1B is this long non-coding RNA, which is induced by P53 and has the capacity to repress MYC locally. Mechanistically, we knew that the effects on MYC are specific because if we look by RNA-seq at steady-state RNA levels, we can highlight MYC as the only gene that's statistically significantly altered. And we also know that PVT1B modulates the transcription of MYC by assessing the nascent transcripts. Furthermore, we have direct evidence that PVT1B RNA plays a role, that PVT RNA production is required for MYC suppression. And we show this by designing antisense oligonucleotides, which are now not disrupting this P53 response element, but knocking down PVT1 transcripts. And so what we see in this case is that, again, whereas in control cells, in the presence of stress, MYC levels drop about 30%. This drop is no longer observed in either of the samples where PVT1B was knocked down using the azo. On the other hand, MYC levels are insensitive to exogenously overexpressed PVT1 isoforms, either 1A or 1B. And this is, of course, suggesting that PVT1B likely acts in cis and not in trans. And this is consistent with the single molecule RNA fish data, which I showed you earlier, which is showing that PVT1B is localized at the MYC locus, so it likely acts locally. Okay, so altogether, this data indicated that production of PVT1B RNA, which is specific to the presence of stress and is due to P53-dependent activation, serves to downregulate MYC levels. And so this is notably an exciting new role for a long non-coding RNA to reverse the effect of a transcriptional activator, in this case P53, into a local repressive signal. But now, to sort of go to the question I started with, what is the functional significance of this regulation? What is the functional significance of these cis-regulatory long non-coding RNAs? And so, kind of just to give you, again, the background, in cancer, MYC is a potent oncogene that promotes cellular proliferation and tumor growth, where P53 is an important tumor suppressor, which through activation of its target genes serves to stall cellular proliferation and limit tumorigenesis. And so given PVT1B's position at the intersection of these two networks, we queried its role in tumorigenesis. And in order to directly get at its role at the organismal level, we decided to employ mouse models of cancer. And so we used the KRAS mouse model of lung adenocarcinoma, this KRAS mouse model, which is crossed to a Cas9 expressing mouse to allow for gene editing. And what makes our model powerful is that we can control tumor initiation via lentiviral delivery that's performed intratracheally in order to infect individual cells in the lung. And by delivering Cre recombinase and a guide RNA, we can both initiate tumorigenesis and perform tumor-specific gene editing. And that happens, of course, in vivo. And so for this experiment, we designed a set of 3 guide RNAs, a negative control targeting tomato, a guide RNA to knock out p53 itself. This is a positive control, which is expected to give rise to bigger tumors. And of course, a guide RNA to mutate the p53 response element that gives rise to PBT1B. And so we histologically performed histological analysis of tumor-bearing lungs at 16 weeks post-tumor initiation. And we observed that both p53 knockout, this was expected, but also the delta RE led to much larger tumors compared to control animals. What was striking in this data, however, was that the tumor burden of these delta RE animals, so again, we're mutating 1 out of 6,000 p53 binding sites in the genome, but the tumor burden of these animals was comparable to the tumor burden of the p53 knockout itself. We actually confirmed this data with also a genetic knockout. And so these results suggest that a really key unanticipated role for PBT1B as the factor responsible for suppressing tumor growth and size downstream of p53, at least in the context of this sort of early stage of tumorigenesis, which this model, mouse model represents. And so to summarize our first story, these findings brought to light the importance of long non-coding RNAs as functional modulators of cancer networks, and revealed a striking finding that long non-coding RNAs, in this case PBT1B, can perform key functions downstream of p53 in cancer. Okay, now that we've shown the physiological importance, for my second story, I would like to switch gears and present an unpublished story in which we queried the linker RNA P21 cis-regulatory walkers to investigate the functional elements of long non-coding RNAs. All right, so to review, as we published a few years back, LinkP21 is a cis-regulatory long non-coding RNA which is involved in reinforcing the binding of HNRNPK and p53 in the promoter of P21, which again pushes its levels past the functional threshold. And so we have used these walkers as an example to study cis-regulatory relationship with the hope that our findings would more broadly apply to other loci as well. And the question that we've asked is, what are the functional elements of long non-coding RNAs that play a role in this cis-regulatory fashion? Okay, so over the past several years, this locus has actually been extensively dissected by a number of independent approaches from our lab and others. And these have included deletion of the entire locus, promoter deletion, mutagenesis of the p53 response element, or azo-mediated knockdown. And in all cases, all of these perturbations led to decreased P21 levels. And so this data convincingly pointed to the presence of a regulatory element in the locus. But the big question is the RNA itself required was unanswered. And so we decided to systematically address this question by developing new genetic tools. And so Lauren Winkler, a graduate student in the lab, first used a well-established approach to ask whether transcription of LinkP21 is required. She inserted the 49 nucleotide premature polyadenylation signal, or PAS, in the first exon of endogenous LinkP21 in the mouse. And then using heterozygous mouse embryonic fibroblasts, which exhibited the expected 50% loss of LinkP21 expression, she asked what was the effect on P21. And consistent with previous findings, she observed a decrease in P21, and therefore the answer was yes. Transcription of LinkP21 is indeed required for P21 expression. Now next, Lauren addressed the question no one has previously been able to answer. Is accumulation of mature LinkP21 required for P21 regulation? So she knew that she would need a new approach, and to that end, she engineered a new allele of LINP-P21 in the mouse, where she inserted in the same position as the PAS a bacterial self-cleaving ribozyme. So Twister is of unknown function in bacteria, but following transcription, it undergoes a quick and efficient RNA-dependent self-cleavage reaction. And so our hope would be that Twister would lead to post-transcriptional degradation of its host transcript, in our case LINP-P21, which would dissociate for the first time the active transcription from the transcript itself. And so Warren again looked at single molecule RNA fish and by quantitative RT-PCR of heterozygous or homozygous MEFs isolated from these mice, which can now contain the Twister insertion LINP-P21. And in both cases, she found that Twister was incredibly efficient at eliminating mature spliced transcript. And so having confirmed that the Twister allele worked as she had hoped, she next looked at P21 levels. And in contrast to the PAS model, she actually found them unchanged. And so she concluded that accumulation of LINP-P21 is not required for P21 regulation. And so, at least in the case of this locus, we are getting closer to defining the functional element. And it appears that transcription, but not accumulation of spliced RNA, contributes to cis regulation. And this is kind of a surprising result, or at least a striking result that perhaps explains the lack of sequence conservation, first and foremost. But in the second place, it also raises questions about the role of RNA sequences and putative RNA structures in the recruitment of protein binding partners. Of course, it's unclear what transcription, what about transcription leads to its role in cis regulation, and we're of course interested in further investigating these questions. Notably, beyond LINP-P21, we now have a set of genetic tools to address the same question at other loci, including PBT1, in order to conclusively determine whether or not long non-coding RNA transcripts are of functional significance. And so I would like to leave you with this slide, which illustrates the idea that long non-coding RNAs are experts of local gene expression regulation in cis. They're experts at creating these local transcriptional environments from the chromosomal all the way to at the gene level, and they're experts at tailoring the expression of their neighbors. And this is, of course, an old idea, but it's definitely not out of date. And actually, I strongly believe that local gene expression regulation by long non-coding RNAs, precisely on this gene-by-gene basis, as I have shown today, may be a key mechanism for both gene activation and repression across multiple transcriptional networks. And when we go back to the idea of cancer, it may also suggest a way to modulate cancer networks therapeutically, such as control of MYC levels, and therefore efforts to understand the functional elements of cis regulation are very important. And so with that, I would like to finish up by acknowledging the people from my lab who've done the work, thank our collaborators and our funding sources. And I'll be happy to take any questions.
Okay, thank you. So once again, we can clap using the reactions button at the bottom if you feel so motivated, and hopefully Nadya can see some hands clapping here. I'm just looking at a few questions that came into the chat room. So from Hubert Salve, we have great talk, 2 questions. What is the molecular mechanism of the PBT1B mediated repression of MYC? And is abolishing PBT1B transcription impact on the MYC regulon expression, considering, although the words are flying up the page, I'm sorry, considering that MYC is a transcription factor? So is that part of the story?
Okay, so to answer your first question, we don't know precisely what's the molecular mechanism, but I should say that we have reasons to believe that in contrast to LINP-P21, PBT1B RNA may matter. And of course, we're going to formally test that using our PAS and Twister insertion models. We are also exploring protein binding partners for PBT1B. And in this case, we actually have this really interesting biological puzzle, which is that PVT1B but not PVT1A can do the job, and they differ by about 200 nucleotides. So this is very exciting sort of biological puzzle. With regard to your second question, PVT1B regulates the transcription of MYC, and it does that so we don't think that necessarily there's a feedback that's going on based on MYC's level as a global transcriptional regulator, because we don't see massive changes of the transcriptome. We see more limited changes.
Great. So Zubing Wu is asking, how do you reconcile the difference between ASO knockdown and the self-cleaving ribozyme? Sorry.
Absolutely. Thank you. It's great to sort of see you. They actually have they leave different 3' ends. So whereas the ASO is a substrate for XRN2 and the torpedo mechanism, that's not the case for Twister. We didn't necessarily have this in mind at the design, but we're actually lucky that this is the case.
Great. So Shengquin Guo is asking, how far is the neighborhood, any chromatin structure or dynamics requirement for this regulation?
So what I can tell you for sure is that in the two loci that I presented, there's evidence for close interaction between the long non-coding RNA and the protein coding gene. Interestingly, we don't necessarily see any evidence that this three-dimensional structure is actually dependent on the long non-coding RNA. In fact, we have strong evidence that it's totally independent, it's pre-existing, it's there. But whether or not the long non-coding RNA takes advantage of this pre-existing structure to have access to its neighbor, we believe that this is likely to be the case.
Okay, so I'm going to ask what I think could be a last question. It's we're getting past 5 o'clock. We can keep asking questions, but I don't want anyone to feel obliged to stay since we're getting a little bit past the hour. So the question is from Carmelo Nucera. Do you have any prediction about a transregulation as well by both of your link RNAs that you're investigating? Do you have any thoughts on this regulation that they could induce in trans?
Sorry, I just lost you for a second. Just a second. Sorry. We don't have any evidence that they can do anything in trans at all. Now, having said that, it's not unreasonable to think that in the context of cancer, an inappropriately expressed long non-coding RNA that goes to a place where it's not supposed to be, like for example, human link P21 is detected in the cytoplasm. And so it's not inconceivable to think that in cancer you may have gain of functions which are not your standard function, but could be the case. And we're very interested down the line in exploring these possibilities as well.
So that's interesting because the next question, and I see Nils Walter has raised his hand, so we'll go to him next. But so there's a question about human mutations associated with link RNAs. Are there more connections like the ones you've been describing?
So, so this is just to make it clear. This is a question whether there's mutations of long non-coding RNAs in cancer.
The specific question is about these two link RNAs, but like in the link RNAs themselves, like.
Yeah. So, so for link P21, the only really mutation that possibly exists actually affects the p53 response element, so which would make sense, of course. Yeah. PVT1, I mean, I can't even, we're just in the beginning of starting to even understand this locus, which again shares this incredible interplay of both oncogenic and apparently tumor suppressive functions. And, I see a massive amount of work down the line in order to dissociate this, but we're developing a number of mouse models and trying to study that. But in human cancer, the locus is altered, rearranged, amplified, or in some other way messed up, in ovarian cancer, up to 30% of something like ovarian cancer. So it's massively deregulated.
Oh, okay, Nils.
Yeah, beautiful talk. I was just wondering, and this is a more philosophical question. So link RNAs, there are lots of these that show these networks of interactions that, because of misregulation, then lead to cancer or exacerbate disease conditions. But at the same time, there's outside the RNA world, there's a large group of people that still do not believe that link RNAs or non-coding RNAs have any function. And so I'm just curious as to how you think about this now that, trying to publish these things. And there are lots of papers of this, of course, and we are all facing this challenge that because there's redundancy, because it's really a large network, you knock one thing out, other things compensate and so forth. It's complicated, right? So.
Yeah, well, so, of course, as a junior investigator in this field, I feel very strongly about this question and about, how the field as a whole pursues this challenge, which is real. I think a major stop to the progress of the field was actually the adoption of techniques that were designed for protein coding genes. And I think that that's why we're thinking of developing these new approaches such as Twister, which actually, you wouldn't really care to do that for a protein coding gene. You just target the ORF with CRISPR. But for long non-coding RNAs, it suddenly makes sense. It could make a big difference. If I were to place a bet, and I guess, you have 200 people here to keep me to it, I would say that whereas LINP-21 was not important, the RNA was irrelevant, my guess would be PVT1 would be relevant. And part that has to do with copy number. LINP-21 is a few copies per cell. PVT1 accumulates in hundreds of copies per focus. And so I think there's a lot of these kind of subtle differences. And I think that this idea that you can just knock out or upregulate or overexpress a long non-coding RNA the way you can do with a protein coding gene is just wrong, and we should move past that and sort of have like tweezer-like approaches that take into account the beauty of RNA biology, really.
Perfect answer. Thanks.
What a great answer. So on that fabulous statement about our beloved molecule, I don't know, should we draw it to a close? There are a few extra questions down there, but what do you think, Nadia? Are you still? Everybody's still here. There's 180 people here. All right, I'll ask a question. Frank Slack wants to know, is there a link between PVT1B in human NSCLC?
Non-small cell lung cancer. That's a great question. Thank you for asking. We're actively exploring that right now. We have to, of course, first of all, look at existing databases and existing data sets, now looking with this new knowledge that an actual alternative isoform exists, which was not previously appreciated. But also we have to be a little bit careful because, that's kind of the advantage of studying the p53 pathway is that, there's quite a bit known about this pathway. The disadvantage is that p53 is inactivated in so many cancers, like right from the start. And so you would expect that PVT1B would not be altered independently of p53. So we have to be very careful of how we conduct these studies. But we're very interested in following up on this question.
I'm just making a note that there's a few messages at the bottom of the chat. You can see the future YouTube channel where you'll find the recordings of these talks. So that'll be handy if you missed any of the points. I've missed them because I've been trying to let people in from the waiting room. But anyway, along with Liz Bilali, who's, yeah. So a final question from Azel Chilek. I wonder, how does the regulation change in the presence of mutant p53? Did you have chance to check the mutant mediated regulation?
We have not. That's going to be the simple answer. I don't know, but we have both the mouse models and the tools to address this, so we should be able to do that.
Great. Okay, I would like to thank everybody for joining us today. I assume that there have been rough patches having to do with getting in the room and so on. And I'm glad that people bore with us and that we've enjoyed these 2 great seminars. And so if there were problems, which we'll iron them out for next time. And I'm just really appreciative that people have shown up and they've shown their enthusiasm for the RNA Collaborative. So I'm looking forward to May 6th. Thank you, everyone.
Great. Thank you.
Carla.
Excellent job, everyone.